So as we're talking about proteins, we've already mentioned that one of the most important functions of proteins is the role they play in metabolism. Because here's the interesting thing, many of the chemical processes that make up metabolism actually occur too slowly to properly sustain life. So we're going to need catalysts to help us along. And when we're dealing with biological processes, our catalyst of choice is often going to be enzymes. Enzymes are just protein catalysts. Enzymes are not consumed by the reaction. They're just tools to help the process along. So kind of think of it like a hammer and nails. If your goal was to hammer a nail into a piece of wood, it is going to take you a very long time to do so. It's going to be an ugly process, it's not going to happen on its own, and it's not going to be easy. But if you had a hammer, you could speed up that process. You could take 100 nails and 100 blocks of wood, and you can use one hammer to put them all together. And at the end of the day, you're still going to have, you're going to have 100 nails hammered into 100 pieces of wood. But you're only going to have one hammer, and the hammer is not affected by the process, it's not broken down by the process, it's not part of the process. Your only goal was to put nails and wood together, and the hammer is just there to kind of help that process along. And enzymes are going to be protein catalysts that speed up that biological or that biochemical process, and they're not going to be consumed in the reaction. Another very important detail about enzymes is that they are also very specific to their chemical reactions, a lot like tools are. So you could use a Phillips head screwdriver to screw 100 Phillips head screws, but you can't use a screwdriver to hammer in a bunch of nails. Or you can't do it effectively. You also can't use a hammer to screw in a bunch of screws. You're going to need a specific tool for that specific reaction, for that specific process. So just like you need a hammer to hammer nails into wood, you're going to need a specific enzyme to drive a specific chemical reaction forward. Another interesting or useful fact about enzymes is that their names often end in the word ase, and they're named for their substrate or what it is they're working on. So for example, the enzyme amylase works on amylose. It breaks down amylose using the enzyme amylase. It breaks starch down into carbohydrates, breaks starch down into glucose in the mouth. So yes, if you ever see that term ACE, it just tells you what it's, or tells you that it's an enzyme. Protease is an enzyme that breaks down proteins. DNase is an enzyme that breaks down DNA. And then another interesting thing to note about enzymes is that they may often need helpers or cofactors that are going to help drive that reaction forward. It helps them run more efficiently. A lot of times we can use vitamins and minerals as cofactors, which is why they're so essential to our diet. Vitamins and minerals most often work by either speeding up or slowing down particular enzymes and helping control that metabolic rate. So they're very important to our diet. Here's a drawing or an example that kind of helps illustrate this process of using an enzyme. In this first diagram, we're using an enzyme to actually break a group up. So we have a single substrate, it's going to bind to the enzyme, and what we form is what's known as an enzyme substrate complex, where the substrate is added to the enzyme, we form a substrate enzyme complex. And then the enzyme allows that substrate to undergo its chemical reaction. In this case, we're going to be breaking it down into its smaller products. So for example, what is the one reaction that we have already mentioned that's used as a degradation reaction for breaking polymers into monomers? Anybody remember what that's called? It's going to be a hydrolysis reaction. Most hydrolysis reactions are going to require hydrolytic enzymes. Enzymes that are going to take the substrate, they're going to take the polymer, bind to the polymer, add a water molecule, and split them up and give us our products, our end products, which would be our one long polymer chain and another monomer, maybe, if this was a hydrolysis reaction for breaking polymers into monomers. And what you'll see is that the enzyme is completely unaffected by this process. It's just going to take that substrate, split it up, send it on its way. Just like you can use a hammer to remove nails from wood. We're going to take that nail hammered into wood, take a nail in wood, use that hammer to split them up, separate them, send them on their way. We can also use enzymes for synthesis reactions. Do you remember what is our synthesis reaction for turning monomers into polymers? It's going to be a dehydration synthesis. So we're going to take two substrates. In this case, it would be one long polymer chain plus a monomer. We're going to add in some enzymes. That enzyme substrate complex is going to form. It's going to produce a single product, so one long polymer chain, and it's going to leave the enzyme completely unaffected. So here's our diagram. If you have any questions about how enzymes work, please feel free to pop into an email or pop into our discussion groups or send me an email, and I'll be sure to cover it again. All right, so one of the things we want to mention, we've talked about what these proteins do. A lot of times these proteins are going to be functioning as enzymes, but what we're going to find is that proteins don't always function effectively, especially if their environment changes. There are a lot of proteins that are going to be found all over the human body that are going to be responsible for certain metabolic reactions. And what we're going to find is that each one of those proteins is going to have the very specific conditions under which it needs to operate. So these enzymes, most of these enzymes are going to be globular proteins. They have bulky three-dimensional shapes. And each of these enzymes have an optimal temperature and an optimal pH under which they can work most efficiently. At high temperatures or extreme pHs, a lot of those bonds that are responsible for that three-dimensional structure, do you remember what those bonds were? They were going to be hydrogen bonds, covalent bonds, ionic bonds, all formed between those amino acids in the sequence. Those bonds will actually break if the temperature gets too high or too low, or if the pH gets too high or too low. We can actually see those bonds break, and when those bonds break, we call this process denaturation. When we lose our three-dimensional structure, the enzyme is no longer going to function as efficiently as it did before. It's not going to function the way it needs to because it doesn't have its three-dimensional shape. And as we saw in this slide, that three-dimensional shape is very important. That site, that active site where the substrate can bind, has a very specific three-dimensional shape that fits that one and only substrate. We then break those substrates apart, send them on their way, and we still have our three-dimensional shape. If we were to lose this little pocket, if we were to suddenly unravel this enzyme into a great big long string of amino acids, we wouldn't have the function anymore. That nature of the enzyme would be changed. This process is called denaturation. Oftentimes, this is caused by changes in the temperature or pH, and unfortunately, denaturation can be fatal in organisms that have too many denatured proteins. They cannot sustain their metabolic process. So the shape of that three-dimensional shape of a protein is very, very important. And so we sort of already talked about denaturation and what happens if we lose that shape, but we're going to talk just a little bit more about the importance of protein folding and protein folding diseases. As we've already mentioned, that three-dimensional shape is very, very important, and we need to make sure that we get that protein folded up correctly. Now, we've already mentioned that proteins will sort of self-assemble based on the amino acid sequence. If we have hydrophobic amino acids, they're going to fold inward. Hydrophilic amino acids are going to stay on the outside. But there are also chaperone proteins that can help proteins fold into their normal shapes. They can also help correct the misfolding of new proteins. They're very important, and defects in chaperone proteins may be playing a role in several human diseases such as Alzheimer's disease and cystic fibrosis. So if these chaperone proteins are defective or not doing their job, we could see misfolded proteins coming off the line. Prions are misfolded proteins that have been implicated in a group of fatal brain diseases known as TSCs. Like mad cow disease is an example of a TSC. And so it's very important that we have correctly folded proteins. Prions are not only misfolded proteins, but they can actually cause other proteins to be misfolded as well. So this is where prion diseases are slightly different than viral diseases or bacterial infections. They're caused by misfolded proteins that actually can cause other proteins to be misfolded as well, and have sort of a chain effect on certain fatal brain diseases or disorders. All right, so that is all we have for our discussion of proteins. Again, if you have any questions, please feel free to send me an email at any time.